what I will present today is a very brief overview, high-level information, only one aspect of sequencing machine. So you can imagine in next-gen sequencing flow, what happens is that we extract DNA from either from vocal, from blood. There is an existing machine where the lab colleagues prepare it, extract it, and then we have some fragments of the DNA that is available. These DNA fragments that are used for an adapter, which kind of like in location or labeling the DNA fragments that is extracted from the samples to suggest that we are interested in looking into these ones. Are they available in the sample or not? That's the question. Then once if they are labeled, and this is the process called library preparation, then we go into the amplification step. So we want to see how often they are expressed, how often they are available in this. If they are much more than that, that means the gene has been expressed quite often. And for that, we use bridging PCR technology. And then one of the technique machine, for instance, that look into individually, one at a time. These individual fragments are here at this stage called reads. And then these fragments is labeled or kind of scanned through and identify which type of nucleotides are they. And that this is summarized into a format that is then transferred to us. And this is only one, as I said, this is only one workflow. There could be many different tiny details that we need to look into. So I introduced the word read to you. So those fragments of the DNA that we are interested in looking, they are called read. And just to give you a resolution of what the reads could look like could be a single nucleotide base pair. So we are talking about one base pair now, to the level of full chromosome, so we can get the whole data set. If I translate into bioinformatics terms or computational terms, let's say, I can get the data, either in kilobytes or in gigabytes. This is the range that we are talking about. When we talk about short reads, then we are around 500 to 1000 KB. But when we are talking about long reads, it's more, and there's also a possibility of longer. All of them has their own questions. So when the questions, which kind of question or what kind of information that we want to extract from them, we can design with the help of the lab colleagues what kind of data that we are interested in, how do we set up the experiments, and so on and so forth. So this is why it is important to understand some background from biology, from the lab. So I talked about omics data. What is omics data? Basically, there are four big omics that quite often is being talked about. So first one is genomics from genome level. Transcriptomic is from transcriptome or RNA. Proteomics from protein, and metabolomics is from metabolome. This four can be classified into two categories based on the techniques that is used to extract this knowledge. One is the sequencing, the one that we have seen so far, and the second one is mass spectrum, which is out of scope of this presentation, so we will not look into that. So let's talk about the genomic and transcriptomic. As I said, that we have to focus on the biological question, what we want to get out of the data, and that can help to extract the knowledge. So one can look into genomic for not only for the information that's stored in genomic, but also interactions to each other. So from DNA to RNA, or from RNA to within RNA. Or there's one more omic style, which is basically epigenomics, which is an additional factor to look how this influences the gene expression, or this influence or genomic changes influence the epigenomics. Okay, so these are the basic omics data that we can get. As I said, I mentioned earlier about genomic data, transcriptomic, proteomics, and metabolomics. But I also mentioned about epigenomics. So when do we start calling it multi-omics or integrative multi-omics? Yeah, this is also a very new and challenging field.
Neugierig? Den kompletten Vortrag finden Sie im Intranet der GMDS. Einfach anmelden und den Menüpunkt Bild und Videomaterial anklicken. Werden Sie Mitglied in der GMDS.